very warm and pleasant good morning to one and all. The smallest organism often have the greatest impact on life. Today, we come together to explore that fascinating world through the lens of one microbiome, one health. It is my great pleasure to welcome all the distinguished guests, esteemed faculty members, project fellows, research scholars, and dear students to today's invited talk on one microbiome, one health, unifying microbes across animals, humans, and ecosystem, organized by the Center for Sustainability and Climate Studies under the Department of Ecology and Environmental Sciences. We are delighted to have your gracious presence, and we sincerely hope that today's session will inspire meaningful discussions and new perspectives on the vital role of microbiomes in shaping the health of our planet. Before we invite our distinguished speaker, let us take a moment to reflect on today's theme. The theme of today's talk, One Microbiome, One Health, reminds us that the health of humans, animals, plants, and the environment is deeply interconnected through these microbial communities. Today's talk will take us into this fascinating and rapidly evolving field, offering valuable insights into how microbiome science is shaping the future of health and sustainability. Now, with immense pleasure, I extend a warm welcome to our respected dignitaries. I respectfully welcome Professor R. Rukmani, Director, Research and Development Cell, Professor Joseph Selvin, Dean, School of Life Sciences, Professor S.M. Sundarapandian, Head, Department of Ecology and Environmental Sciences, Dr. Bharath Prithiviraj, Senior Microbiome Scientist, Reckitt Benckiser, United States of America, Dr. Satish Nalam, Dr. Madhimaran Nadarajan, Associate Professor and Coordinator, Center for Sustainability and Climate Studies, Department of Ecology and Environmental Studies. Before we proceed with today's lecture, let us take a moment to honor our distinguished speaker and express our heartfelt appreciation for accepting our invitation and being with us. I would like to extend a very special welcome to the parents of our esteemed guests who have generously joined us today. Your presence enriches this gathering, and we are truly honored to have you with us. Now, I request our respected Dean, Professor Joseph Selvin, to kindly come forward and facilitate our esteemed speaker, Dr. Bharath Prithiviraj, by presenting him with a ceremonial shawl and a bouquet. It is now my great pleasure to introduce our distinguished speaker for today's session. We are truly delighted to have you with us, sir, Dr. Bharath Prithiviraj, PhD, Fellow of the Royal Society for Public Health, who is currently serving as a Senior Microbiome Scientist in the Microbiome Science platforms at Reckitt Benckiser, United States of America. He earned his master's degree in Life Sciences from Pondicherry University and is one of our distinguished alumni. His work focuses on some of the most exciting areas of modern biology, including microbiome sciences, metagenomics, multiomics, host microbiome interactions. Through this research, he has been successfully bridging fundamental microbiology with real-world applications in consumer and public health, contributing to innovative solutions that have a meaningful impact on society. It is indeed a privilege to have such an accomplished scientist with us today, and we are confident that his insights and experiences will make this session both enriching and inspiring for all of us. Dr. Bharath Prithiviraj, it is now our honor to have you with us, sir. Now I request you to kindly deliver your invited talk on One Microbiome, One Health, unifying microbes across animals, humans, and ecosystems. Sir, the stage is all yours. Thank you.
It's a nostalgic timeline here because 26 years ago I passed out of this department. So it was called School of Life Sciences, of course, and their degree was called MSc in Life Sciences. So now it's called Biochemistry and Molecular Biology, understand? So it's changed, but really nostalgic and also a special day. I mean, August 7th is very special. I'm just going to reflect on it in a bit. But again, thank you to the organizers, Dr. Madhimaran Nadarajan and the Department of Life Sciences, Professor Selvin and the team here. It's really great to come over here and meet the students as well as share some of my experiences over the few years that I've been working in this space, right? Yeah, so let me start with a tribute to Bharat Ratna, Professor M.S. Swaminathan, who is one of my mentors because I did my PhD from the M.S. Swaminathan Research Foundation and he was my doctoral committee member, and I reflect on some of the values that he instilled in generations of researchers, especially around trying to democratize science, making science relevant to grassroots communities and sort of communicate the importance of scientific advances to the betterment of how a common man can utilize or reap the benefits from those. So one of the tenets of how the foundation was propagated was to understand how we could look at microbial capabilities and implement that in local ecosystems. This is also a lot of that what has resonated with me over the years and what I do currently as well, right? And also translating next generation biotechnological tools into deliverable formats for local communities. Again, to Professor MS, I'm forever grateful, and his vision continues to inspire generations to come. With that, let me just go right into the topic that we want to discuss today, right? So we are at a cusp of a very interesting timeline, especially around the data realm, because a lot of what we see right now is AI, and data, and data, right? And as we enter the microbial world, let's take a step back to try and understand the ramifications of how we were perceiving microbes in the decades prior to when this data surges occurred. And now, with the data surge coming through, what is it that we want to look forward to, right? So there's no better topic than trying to address microbial dark matter in the realm of data, because as you know, as we're driving through discoveries and we are trying to integrate technologies, there is the need to understand different approaches that we want to take to try and characterize organisms. So there are approaches such as the conventional way of cultivating organisms, and you want to do it at different pronged approaches, starting with single cell genomics, and you also have the most famous approaches right now, which is metagenomics or transcriptomics, with the influx of what you could do with computational biology, right? And also with AI, you're going to have that repertoire where you could bring in functions, you could push or position into the data insights that you're trying to derive from what you're pushing out in terms of the understanding that you get from these systems. As we think about these different niches or ecosystems, right? So it becomes a multipartite way where you have inputs coming in and also addressing the relevance of what those inputs mean, right? So you have different types of ecosystems, starting with subsurface aquifers or hydrothermal vents, when a lot of the life originated from these areas, right? So I take the example of hydrothermal vents because some of these microbial mats that you see, or some of the organisms that cope with these selection pressures in these ecosystems make those organisms very, very interesting. And lo and behold, not all of them are culturable, and they literally can be classified as dark matter because they are very, very abstract in the way we don't know so much of their preferences for how they can be grown, but we'll have to rely on approaches which are driven by genomics, where you could have these approaches for teasing some of the insights that we could get from them, right? So it kind of has that impact of addressing different systems, starting with something as basic as agriculture, soils, which is a very hyper-diverse environment, and then when you go into host-associated environments like the human gut, where you often see these patterns that are repeating themselves over different geographies, over different ethnic stratifications. So all of those aspects become very, very critical, and it becomes important that they are curated in a way where you can create the understanding for the democratization process. Because yes, human gut microbiome is the most interesting aspect of the microbiome, but at the same time, there's a lot of data surge coming from very specific pockets in the world. Because in the Western Hemisphere, in the Northern American, or even in the European continents, you have more of microbiome data sets that are coming out when you compare it to data sets that are coming out from the African continent, or even the subcontinent, right? So only now there's this catch-up game that's going on, and a lot of the funding that's been coming so far has been on the Western Hemisphere side. But the idea of making microbiome science equitable to all is very, very important. And as stakeholders, I think we hold a responsibility to see that we are able to distribute that in an evenly, scattered fashion. Right? I'll pause at that, but I also want to reflect on these interesting ecosystems that have become more relevant now, because we talk a lot about host-associated, health-associated, but there's also this non-living habitats, which we are mostly engaged in. And now as humans, more than ever, we are spending more time indoors, right? We are in these man-made ecosystems, which we call the built environments, where we're spending more time and are getting more of exposures. Some of them are being driven by patterns such as the pandemic, where they were forced to stay indoors, right? And those also have an effect on how it impacts your health. So I'll touch upon mostly on the built environment side during my talk today. And I'm also going to reflect on some of the metagenomic approaches that have been used so far on how to generate data and make sense of the data, as well as make that data equitable or accessible, in the relevance of the global ecosystem, right? And with these approaches, I just want to also reflect on a study that came out in 2021. This is actually from the genomic profiles of all of Earth's microbiomes. And this was done by the Joint Genome Institute in San Francisco, in California, where they produced this genomic catalog of Earth's microbiomes. So they did this MAG-based approach. This is an assembled genome approach where you actually do a shotgun sequencing or a patchwork of shooting out nucleic acid information from different parts of the genome, and then you have a methodology where you're trying to stitch together information that you get from longer or shorter reads, combinations of those, to make the inference of from where or which habitat you have organisms and of what nature they are. So you have this stratification right from human, which is host associated in terms of the genomes, going down all the way down to the outdoor air or the aerobiome ecosystem, right? So you have different tiers of stratifications here, starting with aquatic, and then there's variation from marine or freshwater, and then the engineered ecosystems, the terrestrial ecosystems, and also the air or the aerobiome, which is actually very much like the netherworld, because the air is very mystical and it's very hard to characterize, right? So in terms of the samples or what you want to derive from biological relevance, it makes it even more challenging. So the reason I bring this up is this was published several years later from the initial study which we did at the University of Colorado Boulder in partnership with JGI as well as with the Oregon National Lab. So Rob Knight, who's my postdoc mentor, my first postdoc mentor, gave me an opportunity to start working on the Earth microbiome projects way back in 2010, where we had this very outlandish goal to characterize microbes from different terrestrial ecosystems or different pockets in the planet, right? So a project like that needs multiple coordination efforts where you need to have standardization in terms of methods that you want to put in place from the lab scale, from the sampling scale, and also from the analysis scale. So we had to surmount through all of these challenges and make the process is reproducible across labs. And this project was collaborators. So the work that started in late 2010 finally got published in 2017 in Nature. So it came out as a consortium paper. It's an open source accessible data set. At that time, we had this as the largest data set in amplicon sequencing. This is a 16S based effort where we characterized the information that we got from these ecosystems globally, where we sourced in information to try and understand what is it that connects us microbially. In this case, what we were looking at was host-associated and free-living ecosystems apart from humans. So we wanted to look at, okay, how is the gut in animal compared to, say, the rhizosphere soils that you find for plants, right? So you could see in this principal coordinate plot the difference that you would kind of observe in the color scattering. So I want to go on to the top figure where I want to compare here in green, right? So if you have deep green, which is representative of the plant corpus or plant rhizosphere, and again, you have the scatter over there where you're looking at the difference between what you would find in saline water or hypersaline ecosystems. So there's a gradient, as you would observe, between the beta diversity or the phylogenetic relationship between these sample types. So what we did was we did an environmental kind of like an ontology tool where we were able to segregate the distribution of phylogenetic information from the microbes specific to the niches that we picked them out from and how diverse they were. So if you could see in deep red in the bottom figure, you had soils. Soils are very diverse in terms of the ecosystems that they represent, the types of microbes that are constitutive of those. So these are some of the large-scale patterns that we were further able to unravel from this data set. And this is actually the starting point of doing things. You know, at that time, given the costs associated with sequencing, the methodologies that you had to put in place to reproduce them, we had to surmount some of those challenges and also get in data sets from different collaborators across the world, right? So you would have someone sending us samples from Africa, from say, the regions around, say, Australia, or, you know, in terms of the Indonesian islands and so on, right? So those are the kinds of diversity in terms of samples we got. But prioritizing the methodologies and also the patterns that we wanted to look at is something that we really appreciated the value or the insight from. So the reason I also bring this figure up is here's a representation of the UniFrac output from the sample types that we see. And in terms of the ones that are host associated, you can see on green plant rhizosphere microbes that really stand out in terms of the unique fraction index. So in terms of how much diversity they represent. And on the free living side, it's also equivalent to the soils where they are more diverse. So plant rhizosphere systems are one of the most efficiently evolved microbiome systems because in terms of selection pressures, these are trained through different evolutionary epochs. And it's one of the most fascinating adaptations that you see day in and day out or over generations, which is actually something you would want to come back to, which kind of asks us to lead to the question as to how are we connected to these soils that coexist with us, right? So there was some of the work that was done post Earth Microbiome Project or the EM and compared with some of the information that we got from human host associated microbiomes. So Rob's team tried to look at what they were doing at that point in time called the American Gut Project. So the methods that we developed for the Earth Microbiome Project at the backbone were the same methods that were used for the American Gut Project, where they were doing this citizen-based study for understanding, especially around the consumers or the individuals, how their dietary responses were in terms of the gut profiles, right? So from there, the question that was asked was, how does this phylogenetically map with each other? So try to compare or think about if you have samples that are coming from the ecosystems around you and you have your own gut samples, how do they match up against each other, right? So this is a beta diversity distribution in terms of the scatter that you see from the diversity, but you do see these regions of overlap between samples representative of human overlaying on the environmental side. This is kind of reflective of the fact that there's this very obvious missing link between environmental microbes that are diminishing in the human gut, which originally were part of the human gut. Because in terms of the hunter-gatherer communities or the remote farming communities, those communities had some of the conserved organisms that were there in nature in the environments that they were living in. But now there is a loss of those microbes because the more and more industrialized we get, the more and more times we spend indoors and not associating ourselves with our surrounding environments, we're going to have a very selective way of diminishing diversity that you see as a metric, which is also reflective as you look at it in this figure here is an overlay of the phylogenetic diversity or the phylogenetic readout from the Human Microbiome Project in red, the American Gut Project in blue, and then in sort of light gray is the EM. And you kind of see that there is these pockets of where the human microbiome or the AGP samples overlay on. There are still some pockets where you could find some of these or like phyla overlapping in the earth microbiome space, right? So these are the kinds of patterns that we further can infer that there is this role or the feeder effect that the environment provides to human host or the human gut, which is actually very relevant in the current times where we're living in, right? So we have what we call the exposome or the environmental exposures that are determining what microbial fate we are met with across interconnected environments that we are exposed to. And here we have different types of vectors that can determine what these environments mean, right? From the human microbiome aspect, we have an interchange with what we share with an animal microbiome or the environmental microbiome that we are part of. And at this, there are different categorizations, starting with antibiotics, which we are overexposed to. And again, there are shifts in the dietary patterns that we are going after, because there's also this westernization phenomenon when it comes to diet. And they've also shown that there is this immigrant effect, especially if you are immigrating from a region which is completely different culturally and even in terms of diet, you're going to have a different adaptation of how you cope with the change to the ecosystem you are moving to. And personally, I've also felt that change when I moved from South Asia or in India to the United States. So those are the kinds of shifts that we see, and we've also seen studies that show the way you're born determines how you cope with in later life to conditions like, autoimmune responses and so on, right? So like C-section delivery stuff and all that. And there's also the pollutant exposure, which is again a feeder effect of the exposome, which again in determining how your overall longevity can be sort of construed. So there's also this point where we want to understand the metrics for environmental chemical inputs and the dietary shift and how that kind of acts as stressors or influences of bridging this divide between these biomes that are interconnected or sometimes maybe disconnected from each other. So as we think about this interconnect, there's always this fact where we want to understand what is it that could be a trigger effect that's going to impact us, and is there something lurking around the corner where we need to be very, very wary about or aware about, right? So that's where we'll go into this area of antimicrobial resistance, which again has not been talked about so much in the recent past as is post-COVID, because COVID was a wake-up call to the entire world where you knew that you are vulnerable to a biological system which can go rogue and takes over in terms of the selection pressure that can be pushed through in a very, very higher frequency. So during that time, there's been a lot of studies especially done around what is the impact that antimicrobial resistance can bring, and the estimations are really staggering. So they're talking about close to 10 million deaths by the year 2050 if antimicrobial resistance or AMR is not tackled right now, because these numbers are way higher than the mortality rates for cancer or for infectious conditions like cholera and diarrheal disease or conditions like diabetes. And this is kind of like the hidden threat which is lurking behind the key pandemic sort of scenarios like COVID-19, which is most talked about and most attended to. The point I want to make here is there's also enough data that's coming out in public domain, starting with there's been a study around trying to understand how usage of antibiotics in agricultural settings is actually creating this massive resistance reservoir. So there's a study that came out in PLOS where they looked at the proportion of abundance of resistance isolates, which is mapped across livestock, in this case poultry or pig or cattle. And in terms of the global prevalence, there's a critical P50 resistant level which is coming on India, especially when you look at these distribution rates around antimicrobial use, around livestock heads, in terms of how much of the distribution of these antibiotics is being used for the livestock alone. And again, humans are 1.4 billion in terms of the population in India, and you also have close to 1 billion in terms of livestock heads that are receiving this particular antimicrobial load, which is actually an alarming thing. There needs to be a guidance of how much of antimicrobials is being used, and in terms of the year-to-year increase, it's an increment of 5%. That is actually a very staggering number that needs to be addressed. So when we talk about such usage in the animal settings, we also want to talk about something in the public health realm, right? So I want to take this example of the controversial novel metallo-beta-lactamase case, which originated in New Delhi, and unfortunately it got tagged on to our capital city, right? So this was in 2008 when a patient in a clinic, an urban clinic in Delhi, was actually screened, and it turns out that he presented this particular mutation, basically the novel beta-lactamase that hydrolyzes some of the carbapenems and penicillin-based antibiotics, which actually creates a detrimental effect in terms of how the patient can be treated. So this transmission got so fast, or so triggered, that it sort of got distributed continentally really rapidly. And as you know, this is coming out of a single transposon, the TN125 transposon, and cross-species horizontal gene transfer events were really accelerated. So much so that they did a study where they found the presence of NDM mutation in the Arctic Circle. And the transmission pathways here is staggering. Starting with the clinical emergence, we are looking at the fueling in terms of environments, which is determined by inadequate sanitation or antibiotic pollution that goes into the groundwater in terms of sewage or the sewage-water interface. And at the same time, once you have this horizontal gene transfer, you're going to have vectors like birds that are transmitting this through their fecal dispersals. So this is a spatial relevance for the mutation that was identified in one region, but there's also this contention around origin point or the source tracking effect of how these mutations were identified. Because the first study that was done by a UK-based group in 2008, and then they found the report that they found the soils in Arctic Circle to have NDM1 years later, literally 24 months later in that particular window. But there's also this need to do a very concerted screening approach or a biosurveillance approach, because in that same timeline in 2010 we had the Commonwealth Games that happened in New Delhi, and several mass gatherings occurred of people travel from different continents, and also some of these studies where they are tracking the plasmid distribution globally is also notwithstanding addressing these facts that you also have had vectors in the form of people traveling from place A to place B, which also needs to be taken care of and also needs to be tracked. So which was not a point, it's again a moot point, especially as we plan surveillance mechanisms or surveillance paradigms for further triggers such as these. The reason I bring that up is with this exposome or the resistome or the presence of resistance in the real world environment, there is a need to track and trace the occurrence and the survival of these organisms that are resistant through the microbes that are coexisting with these chemicals that are in the environment. So you have antibiotics which are relevant for the healthcare settings, but you have antimicrobials which are relevant for the non-healthcare settings in the environments. And one of the key things that is missing right now with the way distribution of antimicrobials or antimicrobial resistance is pervasive is the environmental community transmission aspect. And there is also, as you can see visually here, there is this resistance build up which is happening through the selection pressure and exposure, and you have organisms that are devising new mechanisms in terms of efflux pumps, to take an example, that can create these mutations as well as manifest as biofilm forming groups, which make them further resistant or really hard to remove. And the question I want to ask is, with the existing surveillance systems like the WHA GLASS or the EARS European Net system, are these adequate to monitor environmental community transmission? It is great for clinical surveillance or tracking clinical transmission, but at the same time there is a big lacunae when it comes to environmental transmission, which again plays a key part on understanding how these patterns emerge or how there is an impetus or an increase in the surge of frequency of these infections. That actually leads me to the point of understanding, if you can see even in this illustration, this is a recent study that came out, but I'm just putting it out there for context because you have what they call a hot spot or a cold spot, right? So you have these places like hospitals juxtaposed with wastewater treatment plants. And wastewater treatment plants literally act like feeder systems, because one of the most potent exposures you could have in proximity to a wastewater treatment plant is through the aerosol plumes or the aerosol plumes that you're inhaling. So this is something that's not tracked enough or traced enough, or the methodology that's need to be put in place is also not adequately available universally. Those are the kinds of challenges that we are very interested in, and I've been involved with some of the efforts around aerobiome monitoring, aerobiome methodologies that can be used precisely to track and trace some of these organisms and the spread of thereof, right? So the reason I bring that up is we are in these urban infrastructures, which is actually acting as a living ecosystem. We are living in mega cities, right? And these cities again have metal and concrete and so on, and they are acting as vectors for transmission of microbes. So source sharing is something that's phenomenally universal in these ecosystems. And there's also the need to have global surveillance mechanisms that can reveal a very unique fingerprint of microbes that you would find, which is driven by human activity or the climate paradigm that is governed by different regions in the world. So with that context on the biological layer, I want to introduce you to this project that I've been part of for the past decade called MetaSub. And MetaSub is for the metagenomics and meta-design of subways and urban biomes. So the reason we want to address transport areas in megacities is because this is the space shared by different people from different walks of life. If you think about a city like New York, in fact, this picture has in the background the Grand Central Station in New York, right? And there are people who come in from enormous number of countries. Recently, we had the FIFA Football World Cup. It felt like the whole world descended upon the city. You were literally having people from different continents. So we wanted to understand if there is a source sharing between people coming in and out. Is there a way for us to create a cartographic map, a microbial map, where we can look at spatial patterns of metagenomic information that we could gather from the microbial signals we get, and alongside the information around AMR that goes with it? Maybe it's there, maybe it is not there, right? So this was actually the motivation for this particular project, which got funded by the Gates Foundation back in when it started. And the effort was, again, a little daunting if you think about it, right? So this is a map of sampling that we conducted in 2016 June on a single day in a 24-hour span, starting from Japan all the way out to Alaska on the west coast or western side of the US. You have this timeline snapshot within a month of how many samples were aggregated, right? So it starts with one particular sampling day in June, which coincides with the ocean sampling day. And we collect environmental swabs or samples from these cities where we have metros or subways. So all of the mega cities, New York, London, Tokyo, Rio, were also included in this. So the reason I bring up these other cities is has some relevance. And this is actually one of the snapshots of one particular year. So from 2016 onwards until 2026, in the decade, we've been collecting samples from these surfaces in built environments over the last 10 years. And we've been aggregating this collection, right? So what has happened is we were able to publish this result in Cell in 2021 from the first phase of sampling from 60 cities, just close to 5,000 samples, and which yielded 8 trillion bases in terms of the output. And we were able to narrow down on core microbial taxa that were represented universally in these cities. And we were also able to see patterns of drug resistance, multi-drug resistance across different continents. And putatively, we were also able to see novel bacteria, like close to 700 of bacterial phyla in this case, that were identified, and also putative DNA viruses close to the count of 10,000. So, as you know, viruses are very mystical because what you see in the data is sometimes not fully conclusive, where you also have to do a lot of additional follow-ups that you need to do from that. But this was like the initial snapshot of what we had, which got published, and then you can see this phylogenetic representation from the metagenome data. In terms of the novelty or the known groups, you can see that there's a scatter of novel organisms that you find in these built environments, which have not been seen in any other ecosystem, which is very intriguing, right? And you also have these organisms that have their preference for phenotypic representation. They are, in terms of their preference for being commensal or in terms of being, say, biofilm formers versus not. So there's also that stratification that we were able to notice. Alongside with that, there's also subsequent ways we wanted to understand the data further on trying to understand if this resistance potential in terms of the antibiotic resistance genes that we see from the data set has relevance of distribution across different cities, right? So from different pockets and different countries, are we seeing a rise in terms of the resistance potential? And is it also determined by socioeconomic factors, which is again, the economically challenged countries or nations, whether they have a certain risk factor associated with these strains being lingering there. So we see like there are top resistance classes which are multi-drug or aminoglycoside or MLS markers that are dominating these urban environments. At the same time, the proximity to living in medical being closer to medical centers or having ecosystems or communities living there actually creates this population density, which again dramatically inflates the resistance potential. Again, we want to be able to move on and create, it's not reactive but proactive, but at the same time we are able to do environmental forecasting, right? So for me, the motivation was, how is it that we could use this approach to bring something that we can create insight for our country, right? And this is where the first effort started around Metasub in India. This is the first ever sampling done in a subway or in a metro in our country. And we did this in 2016 in Hyderabad, before the metro was open to the public. We had to go through a lot of resistance to begin with, and we had to go to present to these IAS officers on making them understand what this is all about and try to get samples from here. So the goal being that we wanted to have a baseline to start off from, and then we could go to the next when the public have access to these metros, right? So the challenge here was we were able to collect these baseline samples at that earmarked date in June 2016 that I talked about. But at the same time, we were not able to publish any of the information associated with it because there was a clause from the government saying they were not confident that we would not impact medical tourism in Hyderabad. So we were restricted from talking about this. So although we did the sampling, we also did subsequent sampling during the lockdown. There was still samples available, but again, we had the challenge with not being able to release the information or even further process the information. So that's this challenge that we initially faced. And I want to acknowledge some of my colleagues who are really part of this effort. Narasimha Rao, who's also a Pondicherry University alumnus, and my classmate, and Lakshman Kumar, two of them are from the same university, who were involved with that effort at that point in time, right? So the reason I bring that up is this was the initial effort to try and do this in the built environments, but then it was a failure in a way. We could not push it beyond getting it out. But the lesson learned from there was being persistent enough in a way that if we keep communicating this across in an effective manner to the authorities, you might at some point get the approval to go forward, which actually happened in the case of Chennai. So we were able to move forward and initiate sampling from the Chennai Metro alongside collaboration with IIT Madras, and now we have a data set that's going to come out soon in public domain. So this is actually in revision with the journal M-Sphere. So we did the first sampling from Chennai Metro during the Omicron phase of the pandemic, and we did get the approval to do that. And thanks to the pandemic, the authorities started feeling that the pandemic was a worse devil than AMR. They thought AMR is a lack phenomenon, right? So they were able to, they say like, go ahead and do this. So we were able to do sampling from 12 stations, and what we do is surface swab sampling and also do whole genome metagenomic sequencing from some of these lines that are transporting close to 100 million passengers annually. And some of the patterns that emerged from this study was actually interesting, because you have communities that show up in the data set that are representative of the coastal ecosystem. Obviously, Chennai is a metropolis on the coast. And you also have these organisms that are, resistant in their environments that they are part of, like some of the groups like Pseudomonas or Sphingobium, which are, again, resilient to the pollutants that you find in the urban ecosystems. So these were some things we picked up from the data, as well as there's this human density marker, which is very interesting, because one of the things we found as a recurring theme in these ecosystems is markers associated with human skin, because we leave behind a lot of the microbes that we shed from our skin in these ecosystems in built spaces. So that's something that we picked up as well. And there's also an agricultural link to, again, proximity to agricultural activities, especially in terms of what's coming and impacting in terms of transport, right? So the motivation further is, with what we see is we've been sampling from different areas, like the kiosks, the ticket vending kiosks, the counters, the rods, and we also have some of these samples that are yielding, close to 32 core species with greater than 97% of prevalence. And also you have peripheral species associated with them. So, but what is the take home from here, right? So there's also this paradigm where you see the resistome, which is coming out in urban regions, but at the same time you have the environmental pressure that's further modulating how these resistome parameters are represented. There's the genetic adaptation, which is occurring at the level of transposases or target alterations, which is something that we need to further investigate, because there's this very specific response to disinfection that we employ in different areas. Coming from a company like Reckitt, obviously we make disinfectants too, right? So there's always the need for guided use of disinfectants or chemicals that you want to deploy as antimicrobials. And that's something we also are taking care of. We are working on domains such as those. And there are these latent threats that we also want to be addressing. In the Chennai Metro data set, we also do see some of the latent threats around sulfonamide or rifamycin, where you have the genes associated with these groups that are significantly represented in higher abundances. And that's something that will indicate selection pressure that's kind of influencing that abundance increase, right? And there's this link between the rifamycin resistance, which creates that proximity in terms of how you have the overlap between clinical and urban transit surfaces. If you think about some of the railway stations or metro stations that are in close proximity to the government hospital in Chennai, right? So those are the kinds of proximal areas where you could potentially pick up transmission from healthcare into environments. So that's where we want to be talking about the mechanisms of evasion or replacements, right? So there could be these targets that are getting replaced. And it's actually very important, because selection pressure from these indiscriminate antibiotic use has a way on having that cross-resistance effect meted out to the use of industrial disinfectants, especially, which is forcing these bacteria to adapt quickly in terms of their cyclic change or shift to selection pressure that eventually modifies their molecular structure, which makes them even more potent on adapting to a class of antimicrobial or on an antibiotic molecule. And we further wanted to understand, if bacteria can do this, how about the dark matter? And when you think about the dark matter, there's no better dark matter to talk about than the virome, which is not often talked about, because we always think that, they're there, but they're not part of the tree of life, right? So obviously we care about them. But here we were able to also query that. So we wanted to ask the question as to what is the distribution of viruses, specifically RNA viruses, or the RNA viromes that you would find in these urban or peri-urban regions associated in proximity to the urban sites. And from there, we just published last month in Nature Communication the study where we looked at the diversity and distinct characteristics of global RNA virome in urban and peri-urban environments. So the staggering readouts from here is specific metatranscriptomics that was done, and we had close to 3,000 metatranscriptomes across 31 countries and 102 cities. And in terms of the operational units that were identified, these were close to 50. And 77% of those OTUs from the virome side were completely novel and previously unobserved in the realm of the biological or the microbial world, right? So which is actually, if you think about it, we are literally scratching the surface when it comes to viruses, and we don't know how they operate and what they're doing there. That actually brings the moot point as to what are they doing? What are they doing in there? What is the kind of homogenization effect it is bringing on board? And the more and more we get urbanized, they're selectively collecting populations of these RNA viruses, right? So if you look at this funnel, in the peri-urban reservoirs which are not core urban, you have a distribution of natural environments that have soil and sediments. They still maintain high biodiversity and ecological distinctiveness. And there's also fungi and plants which are dominating the core virome, because viruses associated with plants and fungi are a little different. But when it comes to the urban side, these are viruses that are associated with the human hosts, as well as some of these influences coming in from the habitats like wastewater treatment reservoirs and so on. So there's enough statistical confidence that we can push and sort of interpret that this anthropogenic pressure is diminishing the distinct ecological niche, but at the same time creating a very homogenized way of the virus distribution. And we see these patterns where we can pick up indicators from the virus phyla for built environments, for hospital environments, and terrestrial or trans-based environments, or aquatic reservoirs, right? So in terms of these groups like Duplano, Viricota, I'm not going to read out these phyla, but it's interesting that we have these bioindicators from the virus's side. So some of these indicators, we want to ask what they are doing over there. So we kind of infer that they create this connection between different ecosystems, like the terrestrial ecosystem to the wastewater ecosystem, because wastewater is something that's been more interesting of late, where everybody is monitoring from wastewater to try and understand pandemic strains or distribution, especially when you have an infection spike. But you could also use some of these indicator viruses or RNA virome organisms as something that you can map against what is it that a transit feeder effect can give in terms of how it's distributed back and forth from a wastewater space, right? So this Citrino viricota could be one such organism, which again shows it is something that can affect terrestrial plants and animals. And again, it can also percolate into the urban side of the environment, which is exposed to the environments for humans as well. So there's also this natural reservoir that we came across, which has tremendous phage potential. So interestingly enough, we see that there is an AMR connection to these viromes that we observe, right? Some of the CRISPR spaces that we infer from here, they reveal that there are close to 166 RNA viruses which have the ability to target escape pathogens. So these are the organisms that you could otherwise not clearly or cleanly eradicate with conventional antibiotics. So the messaging here is actually positive in nature, because we have the potential to mine into these phages that we could use as a directed target towards these resistant organisms. And it's about time when we could replicate these approach for fishing novel phages that can be further harnessed for their potential to be used as an antimicrobial. So that's the positive silver lining from these insights that we have. And at the same time, there's obviously this economic footprint, how these pathogens are distributed and how there is a disparity in the nature of these pathogens being distributed. We see that from hospital or transplant samples from countries like Pakistan or Brazil or Argentina for that matter. There's a higher abundance of these vertebrate pathogens, and it's comparable to the Western, like the more economically, prominent cities in the US or so on, like the UK and so on, right? So where it's a little lesser. So that's kind of like a negative correlation that we see, because there's obviously this socioeconomic flip when it comes to these organisms being persistent there. So from the case of what we built here, it's called the urban peri-urban virome atlas, or the UPV atlas. It kind of reveals that each city is not sterile. It's basically a very sort of moving gradient. It's very dynamic in nature, and all of the human activities are going to force, filter and concentrate these viral dark matter entities. And you can see the interconnection here, right? So between the healthcare site to a common transit site to the public space, there's always this interconnection when it comes down to the sewer systems. There's this interconnection of different niches that's going to happen. And you also have the influence that will come from the exposure or the further interaction here. And you also have these phages that can be used as a go-to to balance out if you have a rogue element that's coming up or spiking out in terms of infection spread. So from here, or these kinds of insights, what we want to think about is how could we go towards biologically intelligent smart cities? To have a guided way of understanding how to clean effectively in a very targeted manner. And you have the guidance from a horizon scanning approach, where you want to create these matrix approaches for predicting the propensity of resistance increasing through time and through different niches and at the same time bring in biological control in the form of phages or approaches that you can target towards organisms that are going rogue. And also there's the need for a database surveillance system, which is, again, you have mechanisms like Internet of Things based IoT methodologies, which can be mapped into metagenomic tracking alongside, of course, very complex factors like air quality and traffic monitoring. Air quality, aerobiome is very, very, very complex, but it becomes a very interesting feeder data point for understanding these paradigms, especially with changes in climate and how these microbial shifts occur. So there's also climatic events that can further unravel the cascade of what can be disturbed, right, for ecosystem. We have flooding in coastal regions, we have hurricanes and typhoons and so on, and also the ocean effect like the El Nino phenomenon, which is again determining a lot of, behaviors, starting with even bird movements and so on. So that's actually, again, another factor that we want to be looking at. Geography plays a key part in also furthering these understandings as well. And with the MetaSub initiative, some of the companies that are formed are, one of the ones that I want to highlight here called Bioshare. You guys can go check this out. This came out of some of the data being collected over the last 10 years. And what they have done is having a very interesting way of mapping presence of microbial strains in different cities around the world in these transit spaces, mapped into the local climate for the region from where they were collected. So they have this interface system which is very similar to what the NextStrain database was doing for SARS-CoV-2, but for microbes, for bacteria. So you can go out there, just do check out GeoSeek. The company's name is Bioshare. GeoSeek is the tool which you can again use for this. It's again freely accessible. You can create an account as if you're from the academia. But if you're from the industry, you got to pay a fee. But the founder of this company is one of the guys who started MetaSub. And also there are really remarkable individuals who have put in tremendous effort. I want to acknowledge David Danko, the CTO of the company, and Neem Mohara, so she's the CEO. Both of them came from Chris Mason's lab, who actually is the founder of MetaSub. So there's a lot of interesting work here, potential that we could harness. And I'm also excited at the potential that we could further move to doing sampling for MetaSub, and I want to see if we can bring it to Pondicherry and involve Pondicherry University in this particular journey. And I feel like Pondicherry itself is a very interesting ecosystem because it's so cosmopolitan. You have a floating population coming in every year from Europe and a different set of visitors over the conventional metropolis cities, of course, around India. So we're looking towards such opportunities, and I'm hoping I've brought in some of the excitement around what's being done here and kind of wrap up into this talk here, just going into acknowledgements, right? So there are so many names that there's a lot of people that I need to acknowledge here, starting with my postdoc mentor, Professor Rob Knight, and Mark Hernandez, who I'm forever indebted to for the bioaerosol or environmental engineering training that I received, and Professor Peter Grofman, City University of New York, who's a phenomenal ecosystem ecologist, and my managers from Reckitt Science Platforms and the VP, as well as some of my colleagues in Weill Cornell, Chris Mason, very particularly because he's one of the brains behind MetaSub on some of the funding and the initiatives to bring in people from several countries. Like it's a phenomenal effort where he's brought in researchers from close to 75 countries, right? So, and collaborators in IIT Madras, Karthik Raman, Himanshu, want to acknowledge Manikandan, Professor Manikandan from university. We have a long-standing association since my PhD days, Dr. Damodaran from Mahatma Gandhi, several collaborators, Parnivale here from Lupex, Dr. Madhimaran, and Mr. Santanam from the An Foundation, who was instrumental in the Chennai Metro approvals for doing sampling from there. And several lab colleagues, collaborators globally. Nothing of what we have here would have been possible but for collaboration. And science, being science collaboratively, is the way that you can give back to science as well as to the community that you are part of. So I'm really humbled to be standing on shoulders of these giants. And also, again, I want to end with this quote from APJ Kalam, former president and a visionary. And if you fail, never give up, because fail means first attempt in learning, and end is not the end. In fact, end means effort never dies. If you get no as an answer, remember no means next opportunity. It literally, the lesson I learned from doing some of these efforts around MetaSub, because it's a failure and then we had to go surmount that. And even for me, starting off at Pondicherry University, I never imagined being at a company like Reckitt right now, right? So I did not dream of that 25 years ago, but it's the path that you forge, the opportunities that you take, the collaborations that you create. Obviously, you've had some right people at the right time that you really are grateful for, but I also have to acknowledge my parents at this time because they gave me the freedom to do that, and forever valuable to do that. Thank you so much. I appreciate it. Thank you. Take questions.